Okay, so time to switch into our next speaker, which is Dr. Megan Culler Freeman, coming at us from Pittsburgh as a very newly minted PI of her own group very recently. So congratulations to Dr. Freeman. And thank you for joining us to tell us about innate immune responses to EVD68 in airways and gut. And with that, I will let you take it away.
Thank you so much for having me. I'm delighted to be part of the symposium today and to tell everyone a bit of what I've been working on with enterovirus D68. So my background is at the University of Pittsburgh and the Children's Hospital of Pittsburgh as a pediatric infectious diseases physician and also as a virologist. So I became interested in EVD68 when I took care of patients and families at our hospital in 2018 who had AFM. And it was really part of that process, and also being able to work in the lab of Carolyn Coyne during my fellowship that led me to get into this field. I'll also point out that in the year 2018, Pittsburgh, Pennsylvania actually had the most detections of enterovirus D68 as part of the new vaccine surveillance network study in association with the CDC. So you can see Pittsburgh is that black line at the top that not only almost had the peak here in the first week of September, but really had sustained detections over time. So as a virologist, I wouldn't be doing my job if I didn't tell you a little bit about the background of this viral family. So enteroviruses are small, non-enveloped, positive-stranded RNA viruses, and they actually contains many viruses that you may have heard of before, including Coxsackie viruses, echoviruses, or enteroviruses. And humans are actually the only known reservoir for these infections. They can cause a variety of diseases in humans, anything from a self-limited fever all the way to things like meningitis, endocarditis, or acute flaccid myelitis. They cause up to between 33 and 65% of pediatric acute febrile illnesses, so may very well be the reason that your child gets sent home from daycare. And in the summertime, they can cause over half of hospitalizations for infants who need a rule-out sepsis evaluation, which is when at the Children's Hospital, we evaluate any baby under 8 weeks of age who has a fever. And these can be really devastating pathogens of NICUs. These viruses can cause neonatal sepsis and also death. So enteroviruses are traditionally fecal oral pathogens, and this means that they enter the body through ingestion, and then they go on to have their first round of replication in the gut. They then go through the bloodstream and have primary viremia, where they can land in their secondary tissue site and have another round of replication. And this is where they cause disease, and it varies by virus. So some of these enteroviruses will target the brain, causing encephalitis or paralysis. Some will target the meninges, causing meningitis, the skin, causing things like hand, foot, and mouth disease, or the muscle, in particular heart muscle, causing myocarditis or pericarditis. So enterovirus D68 is a little bit different than these other family members in that it's thought to have its primary replication in the respiratory tract and not in the gut. So I want to just spend a second on why a virus might choose, giving it some qualities there, one entry route versus another. So there are several barriers that your body makes that protects it from infection with different viruses. And this can be everything from viral properties, like the amount of virus a person comes into contact with, the expression of a receptor in a specific tissue type in the body. And, mostly we think about the immune system and how the immune system can make both innate and adaptive barriers that are either kind of general to prevent infections or more specific towards a specific viral or bacterial challenge. So some of these innate immune system barriers can be things like the tight junctions of your skin, the cilia that beat in your respiratory system, mucus that's produced, the act of expulsion through cough or diarrhea to get viruses out of the body, but also things like intrinsic cellular immunity or the interferon and chemical cytokines that cells make that are antiviral in nature after they're infected to prevent the infection from spreading to other cells. We also have non-specific cellular immunity, cells like macrophages that are resident in a tissue and are able to take care of a local infection. Now, there's also the adaptive immune system, and that is when we think about after a vaccine or an original challenge, the body makes very targeted responses both through antibody and specific cells to counter that infection in the future. And I'll leave that arm of the immune system to some of our other panelists this afternoon. So enterovirus D68 is thought to replicate in the respiratory tract. And this is because previous studies of this virus showed that there was reduced replication in acidic environments or at higher temperatures, making the authors conclude that it wouldn't be suitable for replication in the GI tract. But this virus has been isolated in respiratory samples most frequently, but can also be detected in stool and wastewater. So we know that it at least has come into contact with the enteric system. And the study of GI replication has previously been limited by model systems because this is only a human pathogen. So my main question is the enteric tract an unrecognized route of infection for EVD68? And so I'd like to thank Dr. Hodcroft for the wonderful NextStrain platform. And I'm going to show you the viruses that I'll be working with using one of her phylogenetic trees. So if we start over here on the left, you can see that the Freeman strain, or one of the original strains of EVD68, was isolated in 1962 from some children with pneumonia. And then we don't have many sequences between the 60s and the early 2000s. And in the early 2000s is when those previous studies that I was referring to of acid tolerance and temperature tolerance were completed. But then you can see as we move over to 2012, 2014, when we start to notice an uptick in AFM and also an uptick in the circulation of EVD68, that this is associated with not only a greater number of sequences in the repository, but clustering into new clades. So an increase in the genetic diversity of the virus. And for this study, I have used viruses from all of these clades, both from the 2014 outbreak year as well as the 2018 outbreak year of AFM. And I do want to draw your attention to one strain in particular from 2009, down here in blue as part of A1. I'm using that to be representative of the pre-AFM genetic sequence. I chose not to use the Freeman strain just because the way that we maintain viruses over a long period of time is to passage them in cell culture. And you can imagine that if a virus has been passaged in cell culture and lived in the freezer for over half of a century, it may have picked up some adaptations along the way that make it more adapted for cell culture and less representative of a human clinical isolate. So that's why I chose that 2009 strain to be representative of the prior to AFM genotypes. So to tell you a little bit about the cells that I'll be working in, much basic science work with viruses are going to start in cell lines. And these have different attributes in that they can be derived from different tissue types. So I'll be showing you some data in airway cells called Calu-3 cells, and also enteric cells called CACO2 cells. And these exist in 2 dimensions on a dish. They're very easy to work with, but they have some drawbacks in that they're only in 2 dimensions. They're one cell type, and they're actually derived from tumors. So some of the signaling that you might notice in healthy cells is not intact. I will also show you some work from some primary cell lines and organoids, human bronchial epithelials for lungs, and human enteroids or mini guts for the gut model. And these are in 3 dimensions, either in transwells, so at a air-liquid or a liquid-liquid interface, or in a matrigel dome. And these have 3D structures and more preserved signaling pathways and are derived from healthy cells. So let's jump into the data. So can EVD68 utilize lung and intestinal cells for replication? And this is going to start with showing some of that 2-dimensional cell line data. So this is the data from the Calu-3 airway cells. And I'd like to orient you to this viral growth curve because you're going to be seeing a lot of them throughout this talk. So on the y-axis, we're looking at viral titer on a logarithmic scale. And on the x-axis, we have time in hours from 0 to 120 within 20 hours post-infection. This dotted line represents the limit of detection. And what you can see is that kind of the higher the line goes here, the more titer we were able to measure for that virus over time. So the orange virus on the top is really replicating the best, and that is that 2009 pre-AFM strain represented in orange. This experiment was done at 33 degrees to be a little bit cooler and more representative of what you might encounter in the respiratory tract. You can see that all of the isolates that I tested were able to replicate under these conditions. But then when we warm things up to 37, you see a little bit of a different picture. So the orange virus that is from 2009 and replicated so well in that 33-degree model really is right at the limit of detection when we warm things up to 37 degrees. So is not as well adapted for that temperature. However, you will notice that the blue virus here from 2014, a Kentucky strain, is able to replicate well under those conditions. So what about in the gut cells? Here you can see that at 33 degrees in gut cells, that blue 2014 Kentucky isolate is replicating quite well, although you can see that most of the isolates are able to replicate to some degree, but that the orange isolate from 2009 is right there kind of at the bottom. And when we warm things up to 37, things look pretty similar, although overall are able to achieve slightly lower titers at that temperature. So a summary, the main take home is that the 2009 strain really replicated best in lung cells at 33 degrees and not very well under the other conditions. However, some of the more contemporary isolates, in particular, the Kentucky isolate from 2014, was able to replicate both at warmer temperatures and also in gut cells. So what happens when we move things into the primary cell lines? So I'd like to introduce you to the human bronchial epithelial cell line that I use. These are isolated in the airway core at the University of Pittsburgh from lung explants for people that are getting lung transplants. And they're a really amazing cell line in that they are able to both have multiple cell types and they're very functional. They have cilia that beat. You can see the cilia staining in pink on the top panel there. And they're also able to produce mucus, much like the human airway, again, stained in pink, and this PAS stain in the bottom. And these are in three-dimensional transwells at an air-liquid interface. So here I'm showing you growth curves of these viruses when I infect cells at either 33 degrees or 37 degrees. And you can see in the little pictures here that the virus is either being applied to the top, the apical surface, which is similar to if a virus were inhaled and is interacting with the top surface of the cells. And I'm also applying virus to the bottom compartment, which is more similar to if the cells were exposed through the bloodstream or the bottom layer of the cells. And the main take home that you can see here is that the blue virus, the Kentucky virus from 2014, replicates well under all of these conditions. There's another virus that's more contemporary from 2014 from Illinois that also does well in most of these conditions. But surprisingly to me, the 2009 strain prior to AFM did not replicate well under any of these conditions. And the reason that this is surprising is that these human bronchial epithelial cells at 33 degrees should be the most physiologic representation of what's really happening. So just keep that in mind, and I'll address some ideas of why I think is actually happening towards the end of the talk. So for the primary gut model, I use human enteroids, which is a type of organoid or mini organ. These are derived from humans and have multiple cell types, and we can grow them in two different orientations, either in a matrigel dome. You can see kind of the top picture here, what they look like under those conditions. One kind of interesting fact about them is they end up inside out, so the apical surface is on the interior of the enteroids and the basolateral surface is on the exterior, but we can also flatten them and grow them on transwells, and that looks more like what you see in the bottom panel there. And then you have access both to the apical and the basolateral side. So previous work with these enteroids in the Coin lab has shown that they're able to be infected with a variety of different enteroviruses. So you can see in this microscopy panel of enteroids grown in three-dimensional domes. Here is the mock infected panel, so you can just see the nuclei of those enteroids. And then when we go towards the right, you can see green viral staining of Coxsackie virus B, echovirus 11, or enterovirus 71. And this bottom graph is just showing that the enteroids are able to produce a similar amount of titer with echovirus 11 as the traditional cell line. The Coin Lab has also shown that these are also infectable when grown in the transwell system, and access to the apical and basolateral surface is available. So again, you're seeing the mock infected panel here on the left, and then some green viral staining with echovirus 11 and EV71 as you move towards the right. This interestingly had some differences in the capacity for viruses to infect either apically or basolaterally, with enterovirus 71 having a preference for apical infection. Another thing that's really special about these enteroids is they're able to respond to infections. So this is showing some RNA-seq data, and you're looking at a heat map here. So anything that is yellow means that these are upregulated transcripts, and anything that is blue or gray is either downregulated or sort of stable. So you can in particular see for echovirus 11 that you're seeing a lot of upregulation of transcripts involved in the interferon signaling pathways, both interferon stimulated genes as well as many chemokines. And this was true for these enteroids on transwells as well. So this is a really robust model for studying viral infections of the gut. So what happens when we apply enterovirus D68 in this situation? You're starting to sense a theme by now, I think, that the Kentucky isolate from 2014 was able to replicate very well in these enteroids, both at 33 and 37 degrees. And I'm not showing you the data today, but they also, it replicated well when the enteroids were grown on transwells. You do notice that the other isolates, either contemporary or the 2009 strain, did not replicate as well under these conditions, although one of them did actually do a bit better when we moved things to transwells. So in summary, the replication of the 2009 strain pre-AFM was very limited, even in the model that I thought should be the most physiologically representative, these lung cells at 33 degrees. But some of the more contemporary isolates of EBD68 were able to replicate either in warmer conditions or in gut cells and at warmer conditions. So then I wanted to know if lung and intestinal cells were responding differently to EBD68 infection. And I'd like to teach you a little bit about interferon. So type 1 interferon is made up of several family members, including interferon beta and several alphas. It uses receptors IFNAR1 and IFNAR2, which are located all throughout the body. This is thought to be a highly potent, rapid, systemic, and inflammatory signaling pathway. To contrast, type 3 interferon is made up mainly of the interferon lambda family member, which uses the interferon lambda receptor. This is the type of interferon that's used at epithelial barriers and at some immune cells. It's less potent, it's slower, and it's just at those barrier surfaces and is overall less inflammatory to the body. So to make an analogy, if you think about type 1 interferon, it's a bit less controlled, it's hot, it's big, and it's fast, as compared to type 3 interferon, which is much more controlled, a little bit more of a smolder, and in kind of the exact location that you would want it to be to counter a new threat. So I'm going to show you some Luminex data, and this is an assay that looks at 37 different molecules that are secreted by the cells during infection. And you can see all 37 of them there at the top, but I'd really like to draw your attention to the ones in the red box here at the right: interferon lambda 1, interferon lambda 2, and interferon beta. And what you're seeing in the top rectangle is a panel that shows infection with our panel of EBD68 isolates 24 hours post-infection in those primary lung cells. The second rectangle is what happens at 48 hours post-infection. And what you can see that's very striking is that there is a robust in blue, interferon lambda response that starts in the first 24 hours, followed by a interferon beta response that you see at 48 hours post-infection. This was very striking in that these were the only molecules out of this large panel that we were able to detect. And when we compared it to what we saw in the enteroids, it was remarkable in that the gut cells were not really responding with these sorts of interferons at all in response to infection. And we had seen this similarly to some other enteroviruses previously in the Coin lab. If you'll recall, Echovirus 11 was very inflammatory in the enteroids, but enterovirus 71 was much less so. So then I wanted to think back to why that isolate from 2009 didn't replicate well in the primary lung cells at 33 degrees. And I wondered if this robust interferon response was actually limiting the capacity for that virus to replicate. So I wondered if I blocked interferon signaling with a chemical called ruxolitinib, if I would be able to recover the replication of that virus. And so what I'm showing you here is the measurement of interferon beta on the left and interferon lambda 1 in the center. And you're seeing mock infected cells, and then in orange, cells infected with that 2009 strain, and in blue, cells infected with that 2014 Kentucky strain. And on the left of each panel, you're seeing a DMSO or an inert control treated cell. And on the right, you're seeing what happens when we treat with ruxolitinib to stop interferon blockade. So you can see that ruxolitinib does very effectively stop interferon beta and also interferon lambda signaling. And then when I evaluated what happened with viral titer, the blockade of interferon was able to allow for the recovery of replication of that virus from 2009. So I think this actually brings up a couple of interesting questions. Not only is it able to recover that kind of original genotype of virus, but it also suggests that some of the contemporary genotypes of virus might have a mechanism to evade interferon or to counter interferon such that it can really get a foothold and replicate at some of these barrier surfaces. So in conclusion, most EBD68 isolates replicated well in both airway and gut cell lines, although there were some temperature differences. But where we really started to see differences between the strains was in those primary models of airway and gut. The primary airway cells were less permissive to EBD68 infection than I expected, and this is probably because of that strong interferon response that they had that limited replication. Two of the more contemporary isolates replicated well in enteroids, although enteroids did have a limited interferon response to infection. And I think the most important takeaway is that I discovered that contemporary strains have some new attributes. So they tended to have improved replication at warmer temperatures in gut models, and also a potential mechanism of interferon evasion, which just kind of building on what Dr. Hodcroft was talking about is as we're seeing genetic differences in some of these strains, it's probably contributing to the development of some of these changing phenotypes and clinical presentations, although it's unlikely to be the whole story. And it's most likely a combination of something about the virus and something about the host that we are still working to understand. So with that, I have many people to thank, and then I think we'll have some time for some questions. I've been a part of a couple of labs during my time at the University of Pittsburgh, so thanks very much to the Coin Lab and the Williams Lab, as well as the members of the AFM working group, in particular, Gabriella Gulia and Michael Acevedo Woods for their kind of constant communication with me and their work together. I'd like to thank my collaborators in the Airway Corps and at the CDC, and of course, the families who both inspired this work and have worked with me on some other projects. I'd also like to thank my funding, in particular, the Pediatric Infectious Diseases Society, St. Jude's Research Hospital, the Pittsburgh I4Kids, and a recent NIH K08. So I've got a couple of minutes for questions, and if I don't get you now, I'd be happy to connect via email or Twitter. Thanks very much.
Awesome. Thank you, Megan. We just got a question from Mark Abzug, who asks, have genetic defects in interferon pathways been looked for in patients with AFM?
Oh, I love that question. To my knowledge, I don't think that that has been examined at the patient level. I know there has been a fair amount of kind of on the other side of things, the virology side of things, to look at potential mechanisms of how the virus might be able to counter, in particular, interferon beta pathways, mostly through the cleavage of various immune modulators.
Gotcha. And I do think Priya Dugal at Hopkins has been doing some genetic studies on these. So maybe she knows things that we don't know.
I can't wait to see what she finds. I just am not familiar with what the answer is.
Right. And then I think we just have time for this last question from Nicole Warner. Have you looked at other A2 subclade isolates to see if the patterns you've observed with the Kentucky strain exist with close relatives from that same subclade? And actually, can I have you unshare your screen while you tackle that question, and we'll transition?
Absolutely. Probably. No, I haven't is the short answer, but I love that idea.
Good.